I believe that CTC holds the answer to cure cancer. She took the treatment and she responded extremely well. So that was a really touching moment for us, for our team, because something we developed really helped the patients and we saw it happening.
Welcome to Biotech Orbit, where we explore the biotech landscape. I'm your host, Victor Lee. Today's guest is Eric Lin, CEO and founder of Bloodscan. Bloodscan has developed a device that isolates circulating tumor cells from a simple blood sample. What makes their approach unique is that it keeps the entire tumor cell alive. And the process is so simple, it requires no specialized training, making it easier not only for scientists, but for healthcare providers to isolate tumor cells on site. This episode is about the potential of circulating tumor cells in cancer treatment and early detection, and how Bloodscan's technology could reshape the future of diagnostics and personalized care. Before we begin, this podcast is brought to you by Met Productions, a science animation studio where we create animations designed to engage your target audience, whether they are science professionals, patients, or the general public. Visit us at metproductions.media. So Eric, you're living what once was my dream, which is doing applied science and research and then starting your own company. Before we start talking about Labyrinth One, can you tell me how did you get here, like from the start?
Sure, I love magic. I think most people love magic. But science for me is just like another kind of magic. It surprises people with really logical, principles and all kinds of verification, validation, and new exploration. So being a scientist is like being a magician, but has a real impact to the world.
Right. And how did that lead to Labyrinth One? When did that become almost a thing, like PhD or?
So I think I started my research, serious research in high school. During that time, I was in National Taiwan Normal University and with Professor Xianming Hui. She was a professor in the Department of Chemistry, and I was in her lab for about a year doing research in chemistry, synthetic chemistry to be more specific. And I really enjoyed that time. And I think that's the first moment that I really get to enjoy the joy of exploration, of doing experiment, actually. So eventually, when I get to choose what I'm going to study in the college, I picked chemical engineering. And that's where I started later on the foundation of my research. And in college, I did research in microfluidics, targeting using a microfluid device or developing the device to detect volatile organic compound in your breath to detect whether you have some sort of disease.
And then after that, you did your PhD, right?
Yes. After college, I went to the University of Michigan, and that's where I met my PhD mentor, Professor Suita Nagrath. She was really supportive and really, she's a renowned researcher, but at the same time, she was really supportive and really insightful to leading us, all her students, into finding their own path in terms of research and career. And that's where I started to get in touch with our research focus right now, the circulating tumor cells. And so we developed something that's applied microfluidics to isolate those rare cells, the circulating tumor cells.
That's when you started thinking, oh, I can do something with this and apply and start a company. Was that always what you wanted to do?
Not at all, actually. What I really knew about during my PhD was I didn't want to do basic research. Basic research are really important, but I want to know how the research can really help the world, can be applied in the real world. So I was thinking about, during that time, how can this research actually benefit people? Because we don't want to really just stop there for our research results to be just publications or paper, although they are meaningful. But because we were developing a new technology, essentially can potentially change how the cancer care is being operated nowadays. So Startup seems a really straightforward path because then that's the path we can bring our actually research into to be an actual product.
What exactly is it that you're working on? What's the science behind it?
So I think you're asking about what our core products or core technology is, all right? So we developed this product in Bloodscan Biotech. It's called Labyrinth One. It's an automated system for isolating the rare cell, the circulating tumor cells from patients' blood. And what it does is, as a researcher or clinicians or end user, you just need to inject the blood into the system, and it will isolate all those cancer cells automatically for you, all within 15 minutes. And that's really incredible because circulating tumor cells hold so many valuable information, and that's not well studied until still not well studied today. So we're hoping to build this platform that can bring this really otherwise hard to get biomarkers to make them more reachable, more achievable for most of the people or clinicians, patients, researchers, to explore how this can actually change the cancer care system. To bring it to a higher level, our microfluidic device, the Labyrinth chip, it operates purely based on physics principles. It means it doesn't use any antibodies, reagents, or any other kind of labels to capture those rare cells. But instead, we apply fluid dynamics into the microfluid chip so that the cancer cells are aligned and separated by their physical properties. So we keep the recovery rate so high so that we won't miss any rare cells from the bloodstream. But at the same time, we keep them alive because it's very important to have these cells alive even after the isolation process so that we can fully analyze what kind of information that's carried by these rare cells. So for example, their DNA data, their RNA data, their proteomics sequencing data, and more importantly, they are live cells. So we can culture them and we can test drugs on them. So to help people decide what kind of treatment is the best for them. So the initial challenge is how to make this isolation or capture of the circulating tumor cells just make it doable, how to do it. And later on, we'll try to make this technology better. That means we want to capture the most of them because they're so rare. If we miss them, we're probably not getting anything. And we want to keep the cells alive. And so the procedure must be really minimal.
You've been working on this for 6 years, maybe?
No, actually, for business or?
For the whole development of the microfluidic.
We started to develop the technology back in 2011. So that was 14 years so far. And for the first half of it, we were developing the technology itself. So we got our first paper published in 2017, and we got the patent in 2018. And after that, we got almost 20 published papers so far. So after the technology was discovered or invented, we started to think of how to scale it up or how to make it more reachable to more people. And so lots of time, 14 years.
Why CTCs? Why is it important to isolate and collect CTCs?
So from the first day of my research in circulating tumor cells, up to this date, I believe that CTC holds the answer to cure cancer. So now some people will say I'm crazy, but there's a reason I believe in that. So we all know that the early detection of cancer is important because most of the early cancer are curable. But what's really challenging is that the metastasis of them. And what caused the metastasis is actually the circulating tumor cells, because those cells are the rare cells that spread from the primary tumor and get circulated in your bloodstream. And then once they move to a new location, if they find the place is suitable for them to grow, they'll live there and grow as a secondary site. So if we can isolate all those CTCs, study them thoroughly, I believe we can find a real cure to the cancers.
So one is basically finding a cure, which is very important, right? What other applications do you see this technology being used for?
Right, because tumor cells will be shredded from the primary site regularly, every day, actually every minute, every second. So we can actually pick up those rare signals in the early stage of cancer. For example, in one of our clinical study, we can detect up to 85% of the cancer cells in their really early stage, stage 0 to stage 1. So this is extremely important in certain cancer like pancreatic cancer, because most of the times there won't be any symptoms in these patients, and these diseases were not discovered until they are in late stage, say stage 4. So in our data, we can actually distinguish the patients that really has pancreatic cancer, the ones that's healthy, and the ones those are at risk of developing pancreatic cancer.
Are there any real life examples of CTCs benefiting patient treatment?
Yes, of course. Sure. In one of our clinical studies, in this very specific case, it's a patient who was diagnosed with stage 3 non-small cell lung cancer. And during that time, she was just having her third relapse. And it was really devastating for both her and her family and also the clinician. She's been taking so many different kind of treatments or medicines. And so for the clinician, it's really hard to tell what kind of treatment will really work on her. You know? So what we did was, because we were always monitoring this patient, and so during that time, we isolated all her CTCs and cultured them in our lab to get enough of the amount of CTCs because they are so rare. And eventually we tested different kind of medicines, different drugs, and different compounds on the cells we cultured from the patients. And it turned out one of the compounds, the performance is much better than the rest of them. And it was a drug that's still in early stage of development, just phase 1 clinical trial. So we matched the patient to the clinical study program, and she took the treatment, and she responded extremely well and just healthy afterwards. So that was a really touching moment for us, for our team, because something we developed really helped the patients, and we saw it happening. So it was really, really touching for us.
So I see this technology being used a lot for clinical trials.
Yes. Also, we were working with some of the pharma companies because, as I mentioned, local cancers are easily treatable nowadays. So people are finding cure for those remote diseases once they are metastasized. And CTC, as the cause of the metastasis, is actually one of the most difficult targets to cure.
So CTCs are extremely rare.
Yes.
You think that this will help for early detection, right? Do you have any research on that topic?
Yes. We actually had lots of different research data from multiple cancers. So, for example, for non-small cell lung cancer, we can pick up about 85% of the early stage patients, or stage 0 to stage 1 patients. So we can distinguish these patients out from those healthy patients. So it's really a really good screening tool for lung cancer. And the same thing is applicable to liver cancer. And in liver cancer, we detect about 80% of the early stage 0 to stage 1 patients. And also the case I just mentioned about pancreatic cancer, we can distinguish the ones that's healthy, and those are really pancreatic cancer, or those at high risk.
That's a misunderstanding that I had. I thought that CTCs were useful at the later stages, but it seems like it's actually also for early stages, even though they are rare.
Right, because once your tumor, the moment your tumor starts to grow, there are actually blood vessels forming around the tumor, and the bloodstream is, of course, supplying the nutrients for the tumor to grow. But at the same time, the cells start to detach from the tumor itself and goes into the bloodstream.
Makes total sense. So this technology is very much related with liquid biopsies, right?
Yes.
And right now, I believe the routine would be ctDNAs.
Yes.
Is there any advantage of using CTCs instead?
So ctDNA is a great tool because it's more accessible. That means without a specific tool or technology, you can get the plasma from the blood sample and send it to sequencing so that you can get the DNA data from those ctDNA. But the challenge in ctDNA is that those DNA fragments are from dead cells. So that means you are getting delayed information. And also you are limited to DNA data. You are missing the data at the RNA level, at protein level, at the cell level. Because there are lots of treatments that's targeting RNA or protein, and you won't be able to see those data from DNA. But the challenge, this also comes to the challenge for CTC. Why is ctDNA so ahead of time, and why is it already in the clinical practice? It's because that CTCs are really, really hard to get. They are extremely rare, as I mentioned, about 1 in a billion. So unless we have a reliable, consistent tool that can provide high quality of those CTCs, it's really difficult to incorporate CTC in the routine clinical environment. The benefit of using CTC as a biomarker is that it's non-invasive. Normally, if it's a routine test for tissue biopsy, you won't be able to get as much of a sample because it's invasive and it has pose risk to patients. So other kind of imaging, they are expensive or not as approachable. So by just drawing blood, we can monitor the progression of the disease of the patients. They'll be able to know whether their tumors are in further progression or they are responding to the treatment. So monitoring using CTCs is a great use of this biomarker by telling people, okay, if it's a good time that your tumor is growing, or right now your tumor is not really active, so you can take some leave from your treatment to enjoy your life.
So Eric, I know you think big. You don't just want this for a scientist to be using it or in clinical trials. You want it in hospitals.
Right.
What would it take to have Labyrinth One in a hospital?
So first, of course, it's about the regulation. So because we're developing a medical device, it has to abide by all the FDA regulations and also local regulations. So that's one. And second, we need to have enough of clinical data, the validation, to convince the clinicians that this is a useful tool, because essentially we are changing how they are treating the patients or handling the patients. So this has to be done. We need to prove that our technology is reliable with high performance, and those are guaranteed by the FDA regulation. And also, there's a story I can share. Even before we were developing the system, so we did lots of interviews in the States at the beginning of the time when we were thinking about commercialization. So we got one of the feedback that's still is kind of a keystone when we're developing the system, is that if we want people to use a system that's in the hospital routinely, it couldn't be more complicated than pushing a button. And that's exactly how we designed the system. It's really simple to use. You just load in your sample, click start. In 15 minutes, you'll get highly purified CTC.
Oh, 15 minutes.
Yes,
that's all it
15 minutes.
takes.
Yes.
Also, I believe that your CTCs, the enrichment is higher, right? You have higher number of CTCs.
So compared to the gold standard, we capture about 10 times more CTCs.
Oh, that's pretty good.
Yes, that's actually really good.
That's pretty
And on top of that, we keep the cells alive so that we can test drugs on them and we can culture them. We can study their RNA expression.
I think for drug screening, it sounds amazing.
Yes, it is.
And I think this is the right time to talk about the price. Can you tell us how much is it?
Yes, we are setting the price of the system at $50,000 US.
And I think that's very affordable compared to the competition, right?
It is actually. So taking the gold standard for comparison, it's the set price of the gold standard is around $400,000. So most of the other technologies that's similar to us are well over six figures.
Why have it so affordable? Why that price?
We intentionally want to set the price lower so that more people can use it. So as we believe, if we really want to set the standard to change the cancer care system, to make more people use circulating tumor cells in the daily routine of how they handle cancer.
Okay, so at the end of each episode, we have some rapid fire questions. Are you ready?
Yes.
Okay. What's one thing on your desk right now that you can't live without?
It's probably my laptop. I do everything with it.
Do you like stinky tofu?
Oh, for sure. It's my go-to in the night market. If you haven't tried the night market, definitely go for it.
Okay, I'll try it. If you could automate any part of your job, what would it be?
It's probably the meeting with the investors.
Oh, really?
Yes. Yeah, they are really annoying. Sorry. Yes, but you are annoying. Yes, they ask lots of tough questions, but most of the questions I've already answered about 100 times, and I need to follow up with them afterwards. So if AI can automate this process, I would really appreciate it.
Hope they don't get pissed when they watch this. Yeah. What's the most unconventional piece of advice you've been given as an entrepreneur?
Okay, I've heard this one story. It was shared by another entrepreneur. He told me that there will be, of course, it's a metaphor, but there'll be 1,000 reasons along your path to give up of what you're doing as an entrepreneur. If you listen to one of those advice, you'll stop immediately. But as an entrepreneur, you need to listen to a lot of advices because you want to go on the right path. But just don't forget what you're trying to do and don't give up.
Right. If your job was a food, what would it be?
If my job is a food, let me see. I would say it's probably pizza.
Pizza? Yes. Why?
So there are so many ingredients on pizza, right? And there are something you like, you probably don't like that much, but eventually you put them all together and make it delicious. That's pizza. So I'm doing lots of different works every day and trying to make the overall outcome.
All right. And the last question. I know you like social deduction games. Mm-hmm. Could I beat you? If we're in opposite teams, could I figure out if you're the killer or not?
Let's see.
We'll see.
We'll see.
Yeah. That's pretty much it. Thanks a lot, Eric.
Thanks for inviting me.
Yep.